So I'm very interested to look at how by manipulation of metabolic signaling or metabolic pathway we can influence on exhaustion state and somehow restore the function in exhausted T cells. The reason T cells are acquiring exhaustion phase in the cancer patient because once they're entering the tumor environment, they're constantly receiving various signaling including immunosuppressive factors, metabolite stressor, constant stimulation, and accumulation of all of these signalings is adopted T cell to a state to have an exhaustion. They are alive, they can recognize it, but they cannot function properly and kill the cancer cells. We have a different degree of exhaustion. It's not black and white, and it's dependent on the stimulator or signaling T cell receives that the exhaustion state can be varies, but this state is happening, and then each individual of T cells maybe is undergo to exhaustion but not terminally exhausted. So, but all of them after prolonged staying in the tumor environment, they're going to acquire the exhaustion phase. So far in many cancer types already the exhaustion was reported happening among the T cells, and both CD4 and CD8 they're reporting the exhaustion and reaction in the subcutaneously lung, especially melanoma, the most tumor known for exhaustion, but somehow and there are the most tumor type are benefiting from cancer immunotherapy. For example, we have some tumor type like brain, immune cells cannot infiltrate it there, so we don't have exhaustion over the immune system. So one of our main goals of this project is to find the way we can recover or restore the function of T cells in the patient which they're receiving cancer immunotherapy. So ultimately this can be beneficial for patient because their exhaustion phase of T cells can be partially recovered, and that may help the patient to receive the durable response and more effective response. And even for the patient already they don't show response to cancer immunotherapy, maybe this approach can be new potential way to affect on their immune system and restore the immunity. So one of the most challenges us and everyone is facing in this field is about exhaustion are very heterogeneous. T cell exhausted are very heterogeneous, and their behavior can be very much varies between patient to patient. And one of the reasons we wanted to focus on human samples because we want to tackle this heterogeneity. Then other preclinical model like mice model, which is we have one background of genetic, and then it's only one specific angle is going to be explored there. During my PhD at Kyoto, when I was doing with Nobel Prize laureate Tetsuko Honjo, there we are focusing a lot on how metabolism can change the shape of immunity, and that was very exciting for me to observe. By manipulation metabolic pathway we can change the outcome of immune response. And during my postdoc I decided to study this angle, but now exactly on human samples, because I'm very much believing on that in cancer immunotherapy need to be more personalized. We need to be more study on the human derived immune cells than like preclinical model like mouse models.